My name is Ulric, and I worked in the Genoa lab over in the Emerson building. And my project this summer had to do with the function of the RPS6 gene in the Arabidopsis thaliana under normal and cold temperatures. So, more specifically, we wanted to figure out if there is any significant difference in RPS6 A and B deficient plants under those two temperatures, right? So A and B means that there's two copies of this gene, with A being the dominant one, right? So like if it's missing, it's going to have more of an effect. And so we planted a bunch of RPS6B deficient, RPS6A deficient, and wild type seeds in soil as well. And over the course of 11 days, we took pictures and measured them in ImageJ and got their root lengths, right? And so with the root lengths, you can see that there is a significant gap between the wild type as well as the RPS6B. And that is known, that's not new to this study, right? You can see in the petri dishes, there is the wild type under normal conditions almost reaches the bottom of the plate, they're so long, and these ones are about only half the length, right? But if you go over to the cold stress, which was about 10 degrees Celsius for this experiment, they're so close together that with margins of error taken into account, they're essentially the same length, right? And that is not what we expected when we started this experiment. In fact, we expected some sort of maybe overexpression where they ended up above the wild type, or they ended up looking even like a bigger gap. But no, it turns out they eventually ended up about the same. And also within these two graphs, you can see a pattern of RPS6A, RPS6B, and then wild type emerging, right? Which makes sense because it's been pre-established that RPS6A is the dominant copy of the gene. And you can see that as well here in the 2-month-old RPS6 and wild type variants, where it goes AB wild type. However, interestingly, although it's a very small difference with the leaf area of the cold stress RPS6A and B, they are smaller than the wild type, meaning that there is some sort of significant difference there. And then you can see also whether it is because of just this specific sample or because of error in measurement, but I don't think that's what happened. It broke the pattern observed throughout the rest of the experiment where it goes BA wild type, and I find that quite interesting. There's also a noticeable amount of chlorosis going on in the mutants, which is not expressed in the wild type at this point, even under cold stress. So, altogether, what this means is that although the roots don't necessarily show, there is a difference in RPS6 A and B deficient mutants of Arabidopsis, and therefore all plants, given that RPS6 is a gene found in all plants, specifically in their ribosomes. And that could be useful for future studies when trying to make cold-resistant variants of plants. I was a little bit surprised at how non-chaotic it is, and yet how non-organized things seem. You know, when I went into the lab, there's a bunch of stuff everywhere. Like, people were always like in a rush. You know, there's Erlenmeyer flasks that were like half full of some mystery liquid, but everything seemed to be very, very controlled, and no one was ever like frantic. They were all very cool and collected and had a pretty solid plan, it seemed like, of, you know, everything they were going to do that day.